Alright, hello biology students. Today's video we're going to be talking about an important process called transcription. Now, a lot of students have trouble remembering transcription from translation. Easy thing to remember is this word script right in here. Script, S-C-R-I-P-T. Whenever you see that word script, that means you're going to write something because you have to write a script. Well, in the essence of transcription, we have to write a messenger RNA molecule. So, transcription is the process of writing that mRNA. Of course, the M stands for messenger. Now, to me, message and script are kind of the same thing. So that's how I'm able to memorize what transcription is. So that's the process of writing that messenger RNA molecule. Now, let's go, remember here, we're going to be in the DNA, and it's in the nucleus. The DNA never leaves the nucleus because it is so vital to an organism and its survival that we want to keep it protected. So we're going to be in the nucleus right now. Here's one side of my DNA, and of course that's a sugar phosphate backbone. And here's the other side of my sugar phosphate backbone. And of course this is a double helix that has been unwound. And so now we have to put some base pairs in here. So adenine, adenine. If you don't know complementary base pairing, I suggest that you watch my video on complementary base pairing to get a better understanding of what I'm talking about here. So we had some adenines in there in green. Now we're going to add some thymines. Here's a thymine, thymine, thymine. Of course, now we need to add some cytosine. Cytosine. And lastly, some guanine. Let me find another color that we can use here. And we have a guanine and a guanine. Now, of course, now we have to create the other side of the DNA strand and complementary base pair correctly. So in DNA, adenine always bonds to thymine. Okay, so I'm going to put a T there for all the A's I see. T, T. And of course, the thymine bonds to the adenine, so I'm going to put an A for all the T's I see down here. So now we're building the other side of the DNA strand. Cytosine bonds with the guanine. So C and G, and so you get another C and then put a G there. And then of course guanine binds with cytosine. Alright, so now we have fully built our double helix, but it's unwound, of DNA. Now we need to write that message from it. So what happens now is essentially an enzyme attaches itself to the DNA. Okay, so here's the enzyme, and I'm going to talk to you about it here in just a second. This enzyme is called RNA polymerase. The reason I know it's an enzyme is because the ASC at the end there. Anytime you see ASC, that means it's an enzyme. Okay. Now there's a lot more advanced videos out there on the internet to learn about transcription, but my video is just supposed to be a very basic knowledge level to get you off the ground and running. So it's an enzyme, and it's responsible for building the messenger RNA molecule. Okay. So I kind of shouldn't have wrote that there, but we'll work with it, okay? So this RNA polymerase is going to go down the DNA strand, and it's going to read these base pairs. So the first one it's going to go to is that T right here. So I'm just going to draw over some stuff. Okay, so it's going to read that T, and so of course T is what bonds with T, A. And then it's going to go down here and read this C. What bonds with C, G. Now what bonds with A? Now here's the kicker, guys. We're talking about RNA molecules now, so we no longer use thymine. We use uracil. So we write U. I'm just going to finish this out real quick. C. A. Once again, if you're unfamiliar with what I'm doing, you need to watch the video on complementary base pairing. But essentially, the RNA polymerase is going down and reading one side of the DNA strand to create this message. Alright, maybe I should have put so many base pairs, but we'll work through it. Alright, now we have a single-stranded messenger RNA molecule. Okay, so now you see how it's single-stranded. It has a message from the DNA. Now that message, that messenger RNA is going to leave the nucleus, go out into the cytoplasm, and find the ribosome. And that is when we're going to move into translation. I have another video on that you can watch. But for now, that is the end of transcription. That is a very basic understanding of what is going on. I hope you found this helpful, and have a good day.